Hi, my name is Kelly Stecker, and I'm an assistant professor at Utrecht University in the Netherlands in the Department of Mass Spectrometry and Proteomics. And I would like to start by thanking Thermo Scientific for inviting me to speak today and share with you all the work that I'm doing using TMT to help us understand communication dynamics between interacting cells. So one of my research interests is to use mass spectrometry to study protein interactions and protein signaling events that are important for regulating how tumor cells interact with and respond to immune cells. And today I'll share about a method that we've been developing that allows us to directly quantify signaling dynamics between engaged cytotoxic T cells and their tumor targets. And the reason that I'm interested in tumor immune cell communication is that it's really the foundation behind immunotherapy drugs that have taken the field of cancer treatment in an exciting direction with a lot of promise. So immunotherapy drugs such as PD-1, anti-PD-1, these immune checkpoint blockade inhibitors, they act to reinvigorate the immune system to once again target and kill cancer cells by blocking immunosuppressive signaling in the tumor microenvironment. So, however, the problem with immunotherapy drugs is that though the strategy is making a profound difference for some patients, the percentage of patients experiencing resistance to immunotherapy is still quite high. So here I'm showing on the right a graph of progression-free survival curves for metastatic melanoma patients. And on the y-axis, you can see the percent of patients with progression without any tumor growth, and on the x-axis is time, and in blue is anti-PD-1 treatment. And what you can see is that about half the patients right away are resistant to therapy, and from the patients that do respond, about half of them develop secondary resistance within a few months. So resistance to immunotherapy is a big problem in the clinic and requires a lot of primary research to figure out what's going on. And there are many mechanisms driving tumor resistance, but some of them are tumor intrinsic. And by studying what happens when a T cell encounters a tumor cell, we can start to elucidate what cellular mechanisms enable tumor resistance. And furthermore, we can identify druggable targets that can work in combination with existing immunotherapy to improve treatment efficacy. So that's really the goal. And our interest then is to study these two cell types as they engage and interact with each other. So we want to study two different cells during their communication, but this actually presents a technical challenge. So if we try to model the engagement of these two cell types using things like exogenous cytokine treatment or ligand stimulation, we miss the complexity and signaling crosstalk, which is really occurring when these two cell types interact. So here in this video, I'm showing live cell imaging of a T cell in red engaged with a tumor cell in blue. And what you can appreciate from this video is how extensive this communication interface is. Really, a physical attachment is formed between these two cells, and it can be difficult to separate them for analysis for things such as phosphoproteomics, the sensitive measurements. It's difficult to separate this physical interaction. So the question is, how can we measure the individual responses occurring in these two cell types? Because of course, when we homogenize these cells for proteomics, we lose all cellular context. So this is the challenge that we're faced with. And to address this challenge, we adopted a strategy that allows us to measure short-term interactions between two cell types by using stable isotope labeling in our different cell types. So we incorporate Cy-like labels essentially as a barcode to differentiate our two different cell types before we co-culture them together. So how it works in our system is that tumor cells are labeled to completion with arginine and lysine Cy-like labels, heavy labels, and the T cells, which are isolated from healthy patient donors, they are cultured in regular media, so light Cy-like label. And then when we perform our co-culture experiment where we put the T cells and the tumors together, we do this in a media that contains a third intermediate label for co-culturing. So here we have a system where we use Silac labeling to distinguish our different cell types, and when they're engaged with each other in co-culture, newly translated proteins will pick up a third metabolic label. And in this way, we're using Silac labels not for relative quantitation, but solely to identify which piece of information we're looking at in our proteomics data. And so with this approach, we can answer the following questions. Which proteins and phosphorylation events are occurring in T cells, in tumor cells, and in newly translated proteins upon co-culture? And by using these Silac labels, not to make relative ratios as they're normally done, but just to distinguish our three different sources of protein in our co-culture experiment, we next turn to a second quantitative method to quantify the dynamics of these three Silac labels independently. So in our first iteration of this method, we used label-free quantitation to follow these individual Silac labels across our experimental conditions. And when we quantify each Silac label individually using label-free quantitation over time, we can follow the dynamics of proteins and phosphorylation sites in each cell type individually, so in T cells and tumors, and then newly translated proteins upon co-culture engagement. And we called this method hybrid quantitation of Silac labeled interacting cells, HiSeq for short, because here we're using two different quantitative methods to tackle the problem of tracking protein dynamics from two different cell types as they interact. So we established our method, and to test this to study mechanisms of tumor resistance, we employed a matched T cell tumor co-culture system using CD8 positive T cells isolated from healthy donors, and these T cells are transduced with a specific T cell receptor, and they're co-cultured with tumors that express the corresponding matching ligand. So this TCR ligand system fixes the recognition between the two cell types and allows us to study the intrinsic response of different tumor cells upon T cell challenge. So here I'm showing a graph on the right that proves that our T cells do in fact engage with and kill their tumor targets. So on the y-axis is tumor cell count, and in the black line you can see tumor cells in the absence of any T cells, and they continue to grow. And then as there's proportionally more T cells introduced into the system, then we can see that the tumor cells begin to die. So we have a system where we can engage T cells with their targets and induce cytotoxicity, and we use this match system to examine the interaction of T cells with 4 different lung cancer cell lines. This is non-small cell lung cancer, and we took these 4 different tumor cell lines and we co-cultured them with T cells using this match system over a short time course of 0 to 6 hours at 4 different time points. As a control, we included a condition where we had T cells that did not express the TCR, so they were able to interact with the tumor cells but not kill them. So in our experiment in total, we looked at a total of 66 samples in which we measured the proteome and the phosphoproteome. This is co-culturing across the different cell lines with biological replicates over the different time points. We collected this data using data-dependent acquisition and an Overtrap Exploris and a QExactive, and we used MaxQuant to search the data. And we searched it like you'd search a normal Silac run, except we threw out all the ratio data and just extracted the intensities. And what you can see here in the graph of quantified proteins and phosphocytes, and the colors represent the different types of information, tumor, T cell, and newly translated content. You can see that we have far more tumor cell proteins measured and phosphocytes measured than in T cells. So this was not a surprising finding, because we know that tumors are far larger than T cells and they have a lot more protein content. So we expected a bias, but really this was something we weren't exactly happy with, because we're quite undersampling our T cell population. So this is something that that I'm going to come back to later in the talk, and it's something that we were able to address using a TMT approach. So that's a sneak preview for later in the talk. So overall, this is our data set, and the first thing we wanted to ask was, did we actually quantify distinct populations in our mixed cell system? So to ask that question, we extracted all of the proteins which were more abundant in the T cell label, so that's the light side-like label, and did a Go enrichment analysis on these proteins. And what we found, which I'm showing here on the graph, is that we saw a pathway enrichment for T cell specific pathways. So this really let us know that we are preserving cell type specific information with our side-like labels. And it lets us know that our barcoding is working. And next, of course, we have some expectations based on the known biology of the system for what we should see happening when the T cells engage with their tumor targets. So we know that when T cells are activated by their TCR binding to the MHC ligand, we know that T cells release protein molecules such as interferon gamma and granzyme B. And the tumor cells perceive these proteins, these cytokines in the case of interferon gamma, and this sets off a signal transduction event in tumor cells in which a transcription factor, STAT1, becomes phosphorylated and drives the expression of multiple genes, including interferon gamma response factor 1, IRF1. So this protein is, this gene is upregulated in response to T cell attack. So these are things we expect to happen in our co-culture system. And when we looked into our data, we saw exactly this. So if we looked at newly translated proteins, here I'm showing the abundance of different proteins on the y-axis, and the x-axis is time. And the color codes are the 4 different cell lines that we did the co-culture with. So here we see that both granzyme B, interferon gamma, and IRF1 are produced upon co-culture specifically, and importantly, not produced when we do the co-culture with our untransduced T cells. So this is our control in which we are not activating the cytotoxic pathway, so we can see that these proteins are not made. Similarly, if we look in tumor cells at the phosphorylation of STAT1 at the activation site, we see that in all of our 4 cell lines, STAT1 is phosphorylated during co-culture, and this phosphorylation is not occurring when we co-culture our tumors with untransduced T cells. So this lets us know that we can recapitulate the known biology and that our system is working. Finally, as I mentioned, we have 3 pieces of information that we can ask about in our measurements, and that is phosphorylation, protein downregulation, and what proteins are newly synthesized. So if we look at protein downregulation in T cells, we know from previous literature that upon target engagement, T cells downregulate the CD8 protein. And what we can see in our HiSeq data, which I'm showing on the left, is that after 6 hours, we see significant downregulation of CD8 protein in T cells across all of our co-culture conditions. And we validated this downregulation using flow cytometry, so a completely different method. So this, again, shows that we can extract known events with quantitative accuracy in this co-culture system with our HiSeq method. So, of course, after we validated our system, we wanted to probe our data set to find actionable targets that could maybe be used to increase tumor sensitivity to T-cell attack. So what we did was we looked at all of the phosphorylation sites in tumor cells that were significantly changing upon T-cell engagement. And we filtered the data set for phosphorylation sites that were activated across all of the different non-small cell cancer, it's a mouthful, the lung cancer cell lines. And when we cluster this data and we extract the upregulated phosphorylation sites in tumor cells, and we do a Go enrichment analysis, which I'm showing here on the right, we see that several pathways come up, including signaling by Rho GTPases as the second most enriched pathway. And when we looked further into our phosphorylation data, we saw that one of the effector proteins of Rho-RAC GTPase signaling is PAC1, and PAC1 we found was activated by phosphorylation in the tumor cells. So we thought that this would be perhaps an interesting protein that we have identified from our high-seq phosphorylation data as a potential target to see if we can modulate the sensitivity of tumors to T-cell attack. So that's exactly what we did. We returned to our co-culture system. Here, again, we took our T cells matched to tumor cells and we co-cultured them in the presence or absence of PAC1 inhibitor. And what we found is that I'm showing the tumor growth on the graphs on the left, and the y-axis is the cell count for tumors, and the x-axis is time. And in blue, you can see the control condition without any treatment. In red and green, you can see the individual treatment of the PAC1 inhibitor or T-cell treatment. And in purple, you can see the combination of T-cell incubation with PAC1 inhibitor. And in the different cell cancer tumor lines, we see that the combination treatment further sensitizes tumors to T-cell attack. Furthermore, we looked beyond lung cancer and we saw the similar trend in colon cancer, in which the combination treatment further sensitized the tumor cells to T-cell killing, and we saw a similar trend in breast cancer cell lines. So this was quite interesting, and it showed that we could really find a functional target from our mass spec approach. So just to tell you again, to recap what I've said so far, is that we implemented a hybrid quantitative mass spec method, high-seq, to study the short-term signaling dynamics between two different cell types. And in this data, we validated our system, and in this data we saw when T cells engage with tumor cells, they activate Rho-RAC PAC1 signaling, and that PAC1 inhibition further sensitizes tumors to T cell killing. So that was very exciting. But as I mentioned before, we really saw a lot of opportunity to improve our method. So one thing we wanted to do was to improve our sampling depth for all of our cell lines, but particularly for our T cell content, because we are very much undersampling the T cells compared to the tumor cells in our co-culture system. So we wanted to reduce T cell undersampling. We wanted to reduce the amount of missing values in our data sets, because of course that's very problematic to quantifying trends over time and extracting biological patterns. And we also wanted to improve the throughput of our method. So to achieve all of these goals, we turned to a high-seq TMT approach, where we use real-time searching to enable the selection of our target cell types. And I'm going to walk you through this method. So what we did here is, if we return to our original high-seq workflow, where we first metabolically label our tumor cells, and then we co-culture them with T cells in the presence of an intermediate Silec label, we extract these 3 unique Silec signatures and follow their presence over time using LFQ. So here, what we're going to do is we replace, instead of LFQ, we're going to replace it with a TMT real-time search SPS MS3 method. And how we do that is that we do everything at the beginning the same. We do the Silec labeling and the co-culturing, and then we lyse and digest our protein material, and then each co-culture receives a unique TMT label. So this means that each TMT channel represents a unique treatment condition. So that's either time across the co-culturing time points or a biological replicate. Each variable is stored within a TMT channel. And here then, similar as before, when we have our MS1 data, we have the distinct SILAC labels. And so in the MS1 data, we have distinct signatures for our T cell content, our newly translated content, or our tumor content. So just as before, the SILAC labels distinguish the cell types, but now what we have embedded in these signatures is the TMT labels that encompass all of our treatment conditions. And what we can do is take advantage of real-time searching to specifically select isolated precursors for MS3 quantitation. And I will show you how we do that. So what we do is that we, the method is similar as a standard TMT real-time searching approach for MS3, where we isolate a precursor, and then we perform ion trap fragmentation and real-time searching. But in the real-time searching, we specifically set the search criteria for our target SILAC label. So for example, we say, is this peptide a T cell peptide? Does this peptide belong to a T cell protein? If yes, then sequence this, select this for SPS MS3 analysis. So here we are using the real-time search to pull out the cell type that we care about in our sample. And now here I'm showing you the results of this SILAC-specific real-time searching, and what you can see is how much it has improved the sampling of our target cell type. So on the left is the traditional SPS MS3 analysis without the SILAC-specific real-time searching. And here you can see that the tumor cell content is far more abundant than the T cell content, similar as we saw in the LFQ data. Sorry, in the LFQ data, yes. And now on the right, when we do this real-time searching for our light signatures, for our T cell signatures, we all of a sudden have far more, there's a fly, far more quantified T cell proteins. We essentially double the T cell content that we're able to quantify using this real-time searching. And of course, we can decide what we want to target in a real-time search. So if we set up our method so that we specifically choose heavy peptides for MS3 quantitation, and that's tumor targets, then you can see that we boost the amount of tumor targets that we can quantify 2 times. Like I said before, T cell content, we demonstrated we can boost it 2 times. And when we want to target newly translated proteins, we saw an even bigger increase. So basically, the real-time searching allowed us to achieve a 2 to 4-fold increase in our target cell population for quantitation. And already in this data we could find proteins that we before had no information on from T cells. These proteins belonging, for example, to the RAC1, PAC1 signaling pathway that we know is important in this co-culturing cytotoxicity. So that was really exciting. So this really demonstrates the extent of information that we'll be able to extract now that we have more control over our system and control over what we're measuring in the mass spectrometer. Okay. So I want to add a share about something else that we did. So similar to what's done in the SCOPE methodology for single-cell proteomics, we know that we can reserve a TMT channel to boost our target signal. So we tried this for our T cells. So what we did here is that we reserved one of the TMT channels to add T cell content alone, so not a co-culture experiment, but just T cells. And what that enables us to do is to increase the amount of T cell precursor that we see in our MS1 scans. So what I'm showing here is modeling here is in black is the label that's added from our boost channel. And now this precursor T cell peptide is far more abundant and more likely to be selected for MS2 fragmentation. So we've increased the T cell sampling by boosting the channel in the MS1. And if we do this in combination with our real-time searching, in which we preferentially select T cell content, then we have a second method for enriching our target cell population. And what you can see here is how beneficial this approach is. So if we, in this Select TMT experiment, we compared just doing the acquisition as normal with no T cell boost. And for example, in this particular sample, in this 2-hour run, we identified 762 T cell proteins. We quantified 762 T cell proteins. If we add the boost channel, we can increase that to over 2,000 proteins quantified, T cell proteins quantified in our 2-hour run. So this is a 3 times increase in quantification of our target cell population. So that was really exciting. And just to benchmark these values based on our previous Select LFQ approach that we did for our HiSeq method, we basically there across all of our injections, 48 injections at 3-hour runs, in total, we were able to quantify 2,000 T cell proteins. And now with this HiSeq TMT approach using real-time searching and boosting, we're able to quantify more than that in just a 2-hour run. So this is really exciting. And of course, when you get this quantification, you get it across all of your channels, all of your treatment conditions. So really, we'll be able to extract a lot of quantitative trends from these 2,000 proteins. Importantly, to that point, we wanted to prove that our quantitative accuracy is preserved in our Select TMT approach. So what we did here was we created a mixed proteome sample where we had our T cells and our tumor cells, which were heavy labeled and light labeled, and we mixed them at known ratios across the TMT channels. And then we extract back those known ratios and see how accurate the quantitation is based on the expectation. So what you can see on the y-axis is the abundance, and the dotted lines are the expected quantitative values. And the different colors represent the different mass spec methods. So in red, we have the standard MS2 approach. So no real-time searching and no MS3 quantitation, which we know improves the accuracy. In green, we have the standard SPS MS3, which is the benchmark for improved accuracy. And then in blue, we have the real-time searching, where in this case, I'm showing we're specifically searching for heavy peptides for SPS MS3 quantitation. And what you can see is that the MS2 method is not accurate for the quantitation, which is known and to be expected. And to be fair, the complexity of our mixed proteome sample with the Select labels is very high. So the quantitative accuracy is probably even worse than a standard proteome. We see in green that in green and blue that the quantitative accuracy is far greater with our method, and it is not compromised with the real-time searching addition. So it's not quite what it should be for the 10-to-1 ratios, but it's very close. So we are happy with this. Finally, I want to just show that we are able, with this HiSeq TMT approach, that we are able to extract regulation of proteins as we expect. And I'm comparing it here to the HiSeq LFQ data in red, and the HiSeq TMT data is in blue. And on the left, I'm showing again the CD8A protein downregulation, which I showed before. And here in the HiSeq TMT data, you can see we see similar trend, but the variation between the biological replicates is far smaller. This is also true on the tumor side. If we look at the downregulation of granulin, which is another protein that we validated in our data set, in tumor cells, we found that upon co-culture that granulin is downregulated. And if we look at the LFQ data compared to the TMT data, we see that the error bars across the replicates is far smaller. So the precision, the quantitative precision is much higher. And this will allow us to extract statistically significant changes in our data set much better than with the LFQ data. So that's just a preview of the findings that we found from our pilot study with high-seq TMT. And to wrap up, I want to reiterate that using hybrid quantitation, we are able to study short-term signaling dynamics between two different cell types as they interact. And this method has allowed us to identify functional targets in tumor T-cell engagement that we could show, we could manipulate, we could target some proteins from our data set and manipulate the sensitivity of tumors to T-cell challenge. Importantly, the application of real-time searching in our select TMT method has significantly improved the sample coverage for whatever cell type we choose to target. And additionally, boosting target cells with a TMT channel further improves our sequencing depth. And in our method, our quantitative accuracy and precision is maintained. And overall, this is very much an easy approach to probe short-term signaling in intercellular communication. And of course, we used it here in the context of immune T cells and tumor cells. But any cell engagement that you're interested in, this approach would be beneficial. And we're excited to take this in many different directions, including looking at how treatment with different drugs is really manipulating this interface, and we hope to extract which pathways are really important for modulating these responses. With that, I would like to thank, very importantly, the group of people who helped me do this work. So I work closely with Martin Atalar, who's part of the biomolecular mass spectrometry group in Utrecht, along with Albert Heck, who's the leader of the group. There's a very talented postdoc, Alexia Curie, who's been helping with the data analysis. And I have received support from my colleague Tatiana and the rest of the biomolecular mass spectrometry group. Very importantly, our collaborators at the Netherlands Cancer Institute are the ones who developed this matched co-culture system that we use for our study. And I've been doing this work with a very talented PhD student, Sophia, who's part of Daniel Paper's lab. So it's been a really nice collaboration. And I would like to thank Rosa from Thermo Fisher, because she helped us do our first test measurements for our high-seq TMT approach in San Jose on the Ascent. So if you're interested in knowing more, our high-seq LFQ paper is on bioRxiv, and we are working on our high-seq TMT data at the moment. I would just like to end with a disclaimer from Thermo that this work is for research only, and they are not endorsing or promoting what I said. Okay, I look forward to seeing you at the Q&A, and thank you so much for your time.